So thanks, Gauri Shankar. It's a great pleasure to be here. Um, it's been many years since we met. So yeah,
Yes, yes, yes.
um, yeah. So and just also say that um, I haven't done online talks in a long time.
Yes, I
So
pressurized
let's
you
let's see
to.
how this goes.
Okay.
Uh, And so what I'm planning to do is I'm not really giving a research seminar here. Instead, what I wanted to do is to give a broader perspective on the world of bacteria, um, and how we study them today using genomics, and what we have learned in the last 30 years. Well, it's been almost 30 years since the first bacterial genome was sequenced. So we will see what we've learned over the years, and how this might have some role to play in our understanding of um, um, things like bacterial virulence, antimicrobial resistance, and various other biotechnological roles that the bacteria play um, in our world. So I'm quoting this this phrase, unseen majority, from this paper by Whitman and colleagues from the 1990s. Um, it's not, and it refers to the fact that bacteria are the most numerous form of cellular life on the planet. And it's not just a majority. Um, if we just look at some numbers, the number of bacteria on the planet is estimated to be somewhere in the 10 power 29, 10 power 30. The number of insects is believed to be about 10 power 18. Humans, close to 10 to the 10. So this is not just a majority, it's just that pretty much anything other than bacteria are a dot on this large space of cellular beings. They are the oldest forms of cellular life, most possibly, and by far the most dominant forms of life, and capable of an extraordinary range of metabolic and adaptive capabilities that uh, other beings cannot do. And if we, and this is sort of shown in this is a tree of life. This is a phylogenetic tree, and this is a phylogenetic tree made using the sequences of sort of universal genes that are generally found in most organisms on planet. And we see three big blobs here. There is this purple blob at the bottom, and a red and a pink and a green blob at the top. And each of these leaves on the tree correspond to some bacterial or some species on the planet. And the spread of this tree of each of these um, blobs indicate the genetic diversity of that group of organisms. The blue area is bacteria, and of the genetic diversity that we know of on planet, clearly the most dominant form is our bacteria, and eukaryotes are at top, and they're fairly closely related to archaea. And you will this is just one hypothesis for an acceptable phylogenetic tree. There are other complications that arise. There are models which suggest that the eukarya are just a small branch that is leading out of some archaeal branch, and there are also um, the the extraordinary complication in a phylogenetic tree that comes from the idea that eukaryotes came out of a fusion between bacteria and archaea. Then how do you draw those complications aside, assuming this tree is a reasonable approximation of the relationships between organisms on the planet? It's quite clear that the genetic diversity, the known genetic diversity represented by bacteria, is much more than for eukaryotes. And the human knowledge of bacteria goes back a few hundred years, though the bacteria themselves are quite old, 3.5 billion years or so. Um, this is we first heard about bacteria through this guy called Antony von Leeuwenhoek, who lived in the Netherlands in the second half of the 17th century. Um, this is basic. He was by profession a tailor, and this is a facsimile of a tailoring bill that he had made in that time. And this tailor ended up being the foremost microscopist of his time, better than Robert Hooke. And he his microscope was not a compound microscope; it was a simple microscope, and some of his microscopes that have survived to this day can give something like 400x magnification. And using these microscopes, he basically observed things. He observed water, he observed soil, he observed all kinds of things. And he saw what he called as animalcules, which now we know include protozoa, bacteria, and he even discovered anaerobic bacteria 100 years before oxygen was discovered. And this is his in his handwriting. This is the oldest surviving written documentation of Leeuwenhoek's observations. And in his discoveries, you will see that he has very nice descriptions of these green streaks, spirally bound, serpentwise, orderly arranged. Among these streaks, there were very many animalcules. It's the most famous phrase associated with Leeuwenhoek, and he talks about their movements, and he also talks about their sizes in his own way. And his descriptions of these organisms are so extraordinary that about 100 years ago, so, a microbiologist called Clifford Dobell, who translated Leeuwenhoek's writings from Dutch to English, was able to use his descriptions and identify them as the species of protozoa, this species of bacteria, or the other. His descriptions that time were so particular. Okay, so this is in the 1670s, and we've come a long way. And as of a few days ago, if you look at NCBI and search for bacterial genomes, you will find 24 million, 24 lakh, sorry, 24 lakh something genome. So this is there that is listed in NCBI. This could be of varying quality. And of course, now, unlike 20 years ago, not everything that every genome that is available is deposited in NCBI. You have a lot of private and other kinds of public repositories that are there. And if you look at one such thing, which is kind of a nice one for bacteria, you see that there are some 5 lakh something genomes that are there of a certain quality, and which corresponds to something like 1 lakh plus species of bacteria. This is, if we think about it, um, this is uh, quite a significant achievement, um, considering that uh, some 32 years ago, it was not even clear that we will be able to sequence even a single genome completely. And now, we have many, many lakhs of genomes, thanks to primarily to the development of technologies and various other computational approaches that have been developed to analyze and look at these genomes. Now, numbers apart, what have we learned from all these genomes? So at the end of the day, you have the technology to do something, and we keep doing more and more of it. Then one can ask, no, why am I doing all this? What am I getting out of this? And that's what I want to talk about in the next some minutes. So the very first bacterial genome, um, this was to be sequenced. That was not something like E. coli. This was Haemophilus influenzae. It's an interesting organism. The name itself is a misnomer. Haemophilus influenzae does not cause influenza. Obviously, influenza is caused by flu virus. It is just a misnomer caused by an erroneous attribution of influenza to this organism long time ago. So this genome sequenced in 1995 at TIGR, which is now called JCBI in the US, um, uh, came to about 1.8 megabases um, and was predicted to contain about 1,700 coding regions. So Haemophilus influenzae also has an interesting place in the history of molecular biology. We all know that one of the classic experiments that led to the identification of DNA as the genetic material, the experiment by Griffiths and later by Avery, were performed on pneumococcus or streptococcus pneumoniae, as we call it now. And in fact, one of the other organisms in which the phenomenon of transformation, that is the ability of the bacterium to take up DNA from the environment and use that DNA that has been taken up as part of its genetic material. One of the earliest organisms in which this was demonstrated was Haemophilus influenzae. And when this genome was sequenced, the set of genes that are responsible for DNA uptake by this organism were identified. And one more other thing that they talked about is the following: the Haemophilus influenzae version that they sequenced was not pathogenic. It was called RD. So this was not a pathogenic version, and the sequencing the genome showed that if you look at the bottom of this screen, there's this whole bunch of genes called from HIF-A to HIF-E, and these are genes which are known to be involved in virulence in a type B influenza strain. This whole set of genes had basically popped off and did not exist in this strain, which helped explain why this particular variety of Haemophilus influenzae was not pathogenic. So the first genome sequence, therefore, helped rationalize a couple of different properties of this organism, helping make things like genotype-phenotype correlations. From there, now we've said that all these genomes have been sequenced, and are there any properties, general properties that apply across many bacteria that we have learned? The most basic thing that one wants to look at when we are sequencing a genome is how big is the genome, or how large is this organism's genetic material. And this is a density or in a histogram like thing, and on your on the x-axis is the length of the genome in so zero point 0.5 is 5 million and 1 is 10 million bases. And you can see that this distribution is fairly wide. The smallest bacterial genomes that we know of would be as small as about 100 kilobases, and the largest bacterial genomes are somewhere around 12 to 13 megabases. So there are two full orders of magnitude being covered by these genome sizes, and most bacterial genomes are somewhere in that 2 megabase to 5 megabase range, and archaeal genomes are typically smaller than bacterial genomes. And genome size is one thing. There is the other thing is the number of genes that these contain. The number of genes is an indication of the functional capabilities of an organism. And if we were to look at that, you'll see that bacterial genomes have anywhere from say 100 odd genes all the way to 10,000 plus genes. And again, you see that most bacterial genomes have a genome size in the 2000, sorry, the gene count between 2000 to 5000 genes. And if we were to plot genome length that is the number of bases in the contained in the genome, versus gene count, for both bacteria and archaea, you see a very, very tight linear relationship, and which uh, translates to something like one gene every 1,000 bases. So if I'm looking at a 5 MB bacterial genome, I can reasonably confidently say that it will code for maybe a bit under 5,000 genes. And this relationship, as you can see, is very, very tight for both bacteria and archaea. This is very unusual in one sense, in that it's very, very different from genomes of mammals, where you would have heard of the principle of C-value paradox. So if you look at this graph, on the y-axis, sorry, on the x-axis is genome size on a log 10 axis, and on the y-axis is the number of genes. The green dots refer to bacterial genomes, and you can see that this kind of a nice linear relationship that we saw there being shown here. And if you look at the right side of this graph, where I'm looking at large genomes, in particular those of animals in red and plants in blue, and the genome sizes range from 100 million bases plus to something like 10 billion bases, humans, and even larger genomes like fishes and so on. You will see that there is a much greater spread on the x-axis and a very, very shallow spread on the y-axis, which means that genome sizes um, vary over a much larger range than gene count. So many of these genomes will have, say, uh, around 50,000 genes, but they can have anywhere between half a billion bases to 10 billion bases. So, which means that a lot of DNA in these animals and plants, especially, are what being referred to as junk, or that they do not have any known function. And population genetics does suggest reasons why this might be the case, why a bacterial genome is very, very gene dense, whereas a mammalian genome has a lot of genetic material that doesn't seem to have any known function. So in this very fundamental manner, bacterial genomes differ significantly from other genomes. Now, this genome size, gene count are first things that one would want to look at when you're looking at the bacterial genomes. And then the next thing what we want to look at is our genes. Not in terms of just their numbers, but in terms of their identities. Now, if I have a gene in an organism X, how conserved is it? If I take 5,000 other genomes, in how many of these will I find a version of the same gene? Given that there is a lot of similarities in properties of life across different kingdoms of life, one might even say, okay, there may be a lot of similarities between organisms. And it turns out that that is actually not the case. There are very, very, very few genes which seem to be universally conserved. It's at the sequence level. Even if the same function may be conserved, the gene sequence that is responsible for that function may not be conserved. That's exactly what this graph shows. I'm looking at different types of genes, and what fraction of other genomes in which a gene is function. If you were a bar at 1 on the x-axis would refer to the number of such genes which are highly conserved, and a high bar on the left end of this scale would indicate that many genes are very, very poorly conserved. These are bacterial genes looking across bacteria or archaea, and you see that most genes are very, very local, that they are present in a very small lineage of bacteria and not found anywhere else. And if it is found anywhere else, then that it is just very, very, very divergent, that I cannot call it the same gene. So this a range of bacteria represent very, very extreme genetic sequence diversity. And while I was talking about this is across bacteria. I'm comparing, say, a pathogen of humans with, say, cyanobacteria living in the oceans, which are very, very different beings. So one might say, okay, I'm comparing across this vast space of organisms. Why should anything be conserved across? It turns out that even within a species, things may not be very highly conserved. This graph talks about E. coli. And E. coli is an interesting species that represents not only the nice lab strain that we all do our growth curves with in college, but it also represents various types of pathogens of humans, pathogens of birds, pathogens of whatever. And a large number of such E. coli genomes have been sequenced now. And when we are sequencing many genomes of the same species or of the same genus, um, we define two things. One is the core genome, and the other is the pan genome. Now, this is very similar to doing in set theory. If I have two sets and I do an intersection and I do a union, the core genome is essentially the intersection among different sets, each sets being the set of genes present in one bacterial genome. The pan genome is the union across these species. Now, if the species is genetically very, very similar across members of the species, what I would see is that as I add more and more sequences of members of this species, I would just keep finding more and more of the same genes here. The core genome or the common intersection would not be very small, and the pan genome would be closed. That is, as I add more and more sequences, more and more genomes, I do not discover any new genes within that species. And it turns out that is not the case. Um, uh, an, an average E. coli genome would contain about less than 5,000 genes or so, or in that 5,000 gene range. And you see that maybe less than 1,000 of these genes are common across all E. coli. And this could be probably genes involved in translation, transcription, that type of thing. And this soft core, which is a useful thing to define as well, suggests that maybe about 3,000 genes is the soft core. And every time I sequence a new E. coli genome, I end up finding at least one gene that has never before been seen in an E. coli genome. And it's not just one gene, it's several tens of genes is what one finds. So that means as I sequence more and more, I find more and more genetic diversity within E. coli, suggesting that this species has an open pangenome. So the genetic diversity in bacteria doesn't simply only apply to this large scale across this big kingdom of life, but can apply even within species. And now we have the ability to do fairly detailed phylogenetics and try and explain the kinds of processes that lead to these genome size variations. So here is a bunch of E. coli genomes and the phylogenetic tree of these E. coli genomes. These E. coli genomes are named on the right in various W3110, MG1655, and so on. And in this phylogenetic tree, each branch, ignore the pie charts, and you see there are a couple of numbers that says a red number and a black number, and let's focus only on the number that is on top of the line, not at the bottom of the line. So these essentially indicate the number of genes that are gained along that branch, bar the number of genes that are lost along that branch. So you can see that there are certain lineages which undergo massive gene flux or gene gain or gene loss. If you can see towards the right, there is this SD197, the lineage that leads to SD197 has gained some 1700 genes and lost some 2096 genes when compared to its ancestors. So these large macro processes of gene gains and gene losses drive gene content variation, not only in E. coli, but in various other species. So the E. coli is a fairly large genome, 5 MB, but the genome size variation, as we saw at the beginning, is quite vast. They're very, very small bacterial genomes, only few hundred genes. And the second bacterial genome to be sequenced is that of Mycoplasma genitalium, is still the smallest bacterial genome that has been sequenced, bacterial genome that can be maintained in the laboratory, that it is not purely dependent on some host. They're much smaller bacterial genomes as well, but these are endosymbionts. Now this has like some 500 genes or so, and Haemophilus influenza itself, that is considered a fairly minimal bacterial genome. And at the very beginning, they asked, so here I have Mycoplasma genitalium, which is a small bacterial genome, and Haemophilus influenza, smallish bacterial genome. If I look for genes that are common between just these two species, that will probably tell me something about some minimal requirements of cellular life. And on the right, this bar graph shows various gene functions which are represented by the genes that are common to both of these organisms. And as you'd see, things like the most common gene function that is present is translation, and which means that is the absolutely the most conserved gene function. And even smaller than Mycoplasma genitalium are these endosymbiont genomes. And this is a very, very fascinating field, especially if you look up literature on insect endosymbionts, these things like Buchnera, Sulcia, Baumania, and so on. 200 genes, 300 genes, this is all these things can have. And they essentially become machines to supply things that the host doesn't have. For example, these are insects which feed on xylem or phloem, and they don't really get much amino acids primarily. And what these minimal bacterial genomes are basically factories for producing amino acids and vitamins. And you have these interesting cases where you have multiple endosymbionts having formed a relationship with a host, and in which case the two endosymbionts form a perfect or a nearly perfect complementarity in terms of what they produce. If one organism produces a lot of amino acids, the other one would not produce amino acids. For example, in this particular case, the organism at the bottom, Sulcia, is the amino acid factory, whereas the Baumania on top is a vitamin factory. Among the essential amino acids, there is, I think, histidine is the only amino acid which the Sulcia does not produce, and that is the only amino acid that Baumania produces. So these kinds of extreme complementarity and genome reduction in a manner where the endosymbiont becomes a factory for producing something. These are things that one sees in these bacterial genomes. And an extreme case of endosymbiosis, obviously, is the mitochondria, which is essentially a factory for producing energy for eukaryotic cells. And here is a phylogenetic trees of certain segments of the bacterial region, and it highlights three genomes: Buchnera, Rickettsia, and Chlamydia. And all of these are genomes which are reduced in size compared to their ancestors. And these are genomes in which gene loss is a dominant evolutionary force. And if one starts looking at various bacterial genomes, one would see that gene loss seems to be an important evolutionary driver and probably the most common event that happens. And presumably, in the absence of certain processes that allow bacteria to gain gene from the outside, bacterial genomes may start uh, reducing or streamlining in size over time. And at the other extreme of the genome size are these really large bacterial genomes capable of all kinds of crazy things. And here is Pseudomonas, and it has all these interesting metabolic pathways, which you usually don't see. Solvent efflux, mercury resistance, all kinds of nasty solvents this can break down. So these kinds of strange processes are part of certain bacterial kingdoms, bacterial clades with very large genomes. And this is often driven by the phenomenon of horizontal gene transfer that contrasts with vertical transfer means that I'm acquiring DNA from parents. Lateral gene transfer means I'm basically getting genes from somebody, uh, not my parent. And you can see streptomyces, which are among very large bacterial genomes with extraordinary metabolic capabilities, undergo a lot of horizontal gene transfer events even within the clade. And gene acquisition through this horizontal gene transfer based mechanisms is the predominant way by which a bacterial genome grows. So you can imagine some sort of a median bacterial genome, some golden Goldilocks type size, and horizontal gene transfer would be a process which would lead to genome expansion, and gene loss based processes, which can be through all kinds of different processes, would lead to genome compaction. On one side, as the genomes increase, you get bacteria with extraordinary capabilities, high levels of adaptability to all kinds of different environments. On the other hand, as the genomes reduce, the bacteria become more and more dependent on the host and cannot really survive outside their that particular environment to which they have adapted. Now, these gene gains through horizontal transfer turns out to be a critical mechanism by which antibiotic resistance develops among bacteria. And this was really it has been known for some time in various contexts. And this is a very nice study where um, um, they were able to observe greater levels of antibiotic resistance gene transfer between different species of bacteria within the same individual than across individuals. And assuming that if I'm looking at within an individual, that means that the different species of bacteria are in close physical contact and therefore being able to share a DNA and allowing horizontal transfer. And between individuals, you don't really have horizontal gene transfer because of physical barriers. And you see these kinds of antibiotic resistance gene transfers between species more within individuals, more within an individual. Not just antibiotic resistance, also virulence. The most classic example of the gain of virulence by horizontal gene transfer is that of the cholera toxin by Vibrio cholerae. And in this classic paper from 30 years ago, Matthew Walder and George McKellanos, they showed that these bacteriophages are in fact encoded by the cholera toxin genes are in fact encoded by a filamentous bacteriophage, and that the Vibrio cholerae has acquired the cholera toxin genes from bacteriophage DNA integration. And you can see that here when you have a gene of foreign origin, you will see that region being of a slightly different base composition than the surrounding DNA. This little curve at the top shows windows of GC content of the Vibrio cholerae genome, and the region that's there in cyan here, that's where the cholera toxin gene is. And you'll see that in that area you see a big dip in the base composition, whereas most of the rest of the genome operates at some higher level. So these kinds of horizontal transfers have played a role in broadly in the evolution of Vibrio cholerae, starting from an ancestral Vibrio cholerae down to what is known as the seventh pandemic or classical and Eltor varieties of Vibrio cholerae, which involves acquisition of first um, 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 a certain receptor to which the bacteriophage carrying cholera toxin can bind to, and then the acquisition of the cholera toxin, and then through recombination, a switch in state of the cholera toxin from a classical type to an Eltor type, and various other recombination and horizontal transfer processes lead to development of different strains of pathogens in different stages in the pandemic. And while we're talking about horizontal gene transfer, it's kind of popular to talk about these in the context of virulence, but it turns out that even fundamental metabolism can be horizontally transferred. Something as fundamental as components of bacterial photosynthesis may have been transferred around by bacteriophages. And this is what you see here. This is a gene tree of certain genes involved in cyanobacterial photosynthesis, and you will see that there are genes encoded within bacteriophages being evolutionarily very closely related to bacterial um, genes, so which suggests that there might have been derivation of one from the other, and uh, acquisition of these pathways um, by horizontal transfer. So bacterial evolution does involve these macro processes like gene acquisition by horizontal transfer and gene losses. But at the end of the day, there is also mutations. This is just a mutation-based phylogenetic tree of a bunch of E. coli strains that were derived by evolution in the laboratory, evolution over about 50,000 generations. On the left, this dot you see is the ancestor, and going from there, various lineages have emerged, coming with their own mutations. And the phylogenetic tree that we started off with is essentially built based on mutations, and they have nothing to do with horizontal transfer and so on. And then the question arises, when I'm looking at a phylogenetic tree, how do I incorporate these other processes that are not just simple mutations? And this is one more example of E. coli populations accumulating mutations over time when they're maintained in stationary phase in the laboratory for a month without any nutrition. And this is an interesting phenomenon by which stationary phase is not really stationary, but is a very, very dynamic phase where you have mini growth curves of different variants of bacteria coming up and going down, and overall resulting in an increased genetic diversity within the population. And these mutations obviously also have roles to play in antibiotic resistance. We talked about antibiotic resistance through horizontal transfer, and that is very, very commonly seen in things like E. coli, Klebsiella, the so-called escape pathogens. And on the other hand, in Mycobacterium tuberculosis, where antibiotic resistance is also again a very large problem, this resistance evolves through mutations. For example, here are various mutations that are seen in various strains, and these different colors indicate uh, mutations causing resistance to different antibiotics. For example, you see in that dark blue thing, the second one from the left, mutations in RPOB, which is the a catalytic subunit of the RNA polymerase, which is the target of first-line antibiotic rifampicin, and mutations there will confer resistance to that, and so on and so forth. So these are some of the things that we've learned from 30 years of sequencing, annotating, and understanding bacterial genomes. And this is all very technical. And to do that would require a more comprehensive workshop than a brief talk of this kind. But this essentially means that we should learn how to, once we have genome sequences, how do we assemble these short sequence fragments into a complete or near complete genome? And once I have a complete genome, how do we annotate these genomes? And once I have annotated genomes, I have to understand some fundamental principles of molecular evolution, and use that to be able to perform comparative genomics and build phylogenetic trees and so on. How do I identify orthologs, paralogs? How do I make a phylogenetic tree for a set of organisms that I'm interested in? And once this is established, and have some basic statistics and hypothesis testing skills in the head, and then comes the hardest part, which is to ask interesting questions of these data, and look for answers in the data. And I will not really talk about the methods that are used for all of this, because that's not something I can really do justice to in a short period of time. So on that note, I will stop, and I'll be happy to take questions.
Yeah, it's a great talk, Ashwin.
Thank you.
Yeah, so the session is open for questions. So anyone can and can ask a questions. You can type it in the chat, or I'll unmute. So you can unmute and then you can ask a questions. Everyone has gone for their dinner or so. No one is. Yeah. So there's a question. Uh, uh, what regulatory mechanisms enable bacteria to alter their metabolic pathways when
Yeah.
transitioning from aerobic environment to anaerobic environment?
So yeah, there are all kinds of things. If you look at E. coli, there are various mechanisms. Um, there are two fairly global regulators of transcription in E. coli, and one is called FNR, which is essentially a protein with an FES cluster, and the FES cluster's redox state determines whether the transcription factor is active or not, and depending on that status, it would activate or repress the expression of a whole bunch of genes. For example, keep the TCA cycle on only during aerobic conditions, and so on. And different bacteria have different mechanisms. So underscoring the diversity. This FNR family is, though it's very important in E. coli for this kind of thing, is not very highly conserved outside of related bacteria. If you look at some other bacteria, you have some other mechanism for doing this. And the common way by which this will happen would be through a transcription factor, sensing redox state in some manner, probably an FES cluster status. So that's typically how it goes, but the details can vary depending on the system, depending on the organism.
The next question, Ashwin. How
Yeah.
the genetic sequencing or mapping can be exploited for human disease diagnostics and treatment? Please give some insights by Gandhiraj.
Yeah. So the most visible example that we had of genome sequencing contributing in some way to health was during the COVID pandemic, not bacteria, of course, that is a virus, but the same principles apply. Understanding genome variation helped to know what strains are out there, and whether new versions are coming up and so on. um, and from in the bacterial side as well, there's been what's called the genomic epidemiology, where one sequences at a very high resolution many genomes that are part of the same epidemic, for example, and then try to trace their ancestry. And there have been a few fairly popular examples where such experiments helped find the source of a local epidemic to one individual. That individual was the person who was carrying the founder strain, and once that individual was found and turned out to be a carrier and was isolated, the epidemic was stopped. So that's been one uh, direction in which genome sequencing has helped, which is primarily in epidemiology. Um, One can also, in theory, argue that you can use genome sequencing, annotation, etc., to develop new antibiotics and so on. But in practice, how useful that has been is, I think, I would say, is up for debate, because we still make antibiotics through screening efforts primarily. Though there are people who are trying to use genome type information to predict drug targets, etc. In theory, yes, it might work, but in practice, I don't know.
A question from my side, like, what is the time period it takes to evolve? Like, how fast it changes the genome? Like virus, it changes pretty fast. How
Yeah.
fast? Yeah.
So, okay, so one has to look at this in first case, is the genome itself changing, or there is already some random distribution of mutations and selection operates to push one variant count above the other. So that aside, you can actually take E. coli, okay, just a simple lab experiment, right? You take lab E. coli, grow it in the presence of rifampicin, low concentration of rifampicin, okay, not things that will immediately kill high concentration, low concentration of rifampicin, and one day later, that population you will find will be resistant to rifampicin genetically.
Okay. Okay.
So it
Okay.
can be very, very fast.
Okay. That's great. So there's a question from T. Gopalakrishnan. Can you just go through it because it's pretty big? So in the
It's
chat,
in chat.
it's in
Uh,
chat.
what
Uh,
specific quorum sensing mechanisms with the different microbial available signal, AV, UV radiation, and so on. Nice. So quorum sensing is, obviously, it is one of the many regulatory mechanisms, and this is one process by which bacteria sense themselves, their own concentration is what they are sensing. And typically, one will find that um, these quorum sensing mechanisms would regulate things like biofilm formation. There is a very, very high concentration of bacteria of the same type in a small area. Okay, so we can form a biofilm type structure there. And in some cases, this can also regulate, okay, the release of, say, some toxin in a clinical context. So those kinds of things have been known to be regulated by quorum sensing. And one of the classical discoveries of quorum sensing is in the squid and that light sensing organ, where the light comes from bacteria. And emission of the gene responsible for that light emission is triggered by quorum sensing mechanism. And UV radiation and other things. E. coli, we talk about things like SOS response and so on, which primarily tries to sort out DNA damage, and that's a very very vast field. And we're now starting to realize that many of the paradigms that were established by studies in E. coli do not necessarily apply to other bacteria. It's a lot of diversity in mechanisms by which these things happen. Again, with desiccation, one common thing that many organisms use, so here it's the other side, that where you see similar mechanisms in bacteria, in yeast, in C. elegans, and so on, is to produce a lot of trehalose, the disaccharide trehalose, very very abundant sugar, and that seems to be helping these organisms protect themselves against various stresses, including desiccation. And moving between different habitats, it again comes down to what are the specifics of that habitat and what that organism is sensing. And if the organism is able to sense certain change in that habitat, then it's likely that it will trigger some kind of a transcriptional regulatory mechanism downstream, which will lead to some different gene expression state, which is presumably more adapted to that condition. And again, every organism and every habitat will have its own mechanisms. And we've studied these in a lot in E. coli and in some model organisms, but this our understanding of these processes, this we should be very clear. I've told about this 5 lakh genomes, 24 lakh genomes, and so on. We have these large numbers because it's very, very easy to sequence bacterial genomes now, right? You can pretty much do it in a day. You can probably do many in a day. So we can do the easy things. The hard part is to work out all these mechanisms, figure out what it might be doing, all these, answering these questions across a large number of bacteria. That is hard, and we are nowhere close to matching the data that we've produced in terms of genome sequences. We know a lot in some model organisms. Beyond that, we know next to nothing.
Ashwin. So one from Saravanan Perisami, how to predict variation in microbial pathogenic biofilms in clinical context?
Pathogen as in genetic variation.
Yes.
I would say you cannot really predict. You have to sequence the genomes, and once you have the sequences, there are several different ways by which you can try and quantify the genetic variation that is there between different isolates or different locations or something like that. So it's basically sequencing genomes. Um, and from a logistic standpoint, it's relatively inexpensive now, especially if you are going to sequence several at the same time. You can just put multiplex them, and then it gets done very quickly.
This last question. So it will be role of bacteria to help in prevention of climate change. Any metabolic approach recently? By Yashoda.
Yeah, these are big questions. There is what aspect of climate change? No, it's difficult to answer. There are things like climate change is very, very multifactorial, right? It is not just temperature. There are lots of different things, pollution, everything contributes to this. um, and um, and bacterial metabolic capabilities are such that you can, if we can potentially develop ways to use them to clean things up, for example. And one of the most extreme things that I've heard of are bacteria which can even clean up um, um, wastes in nuclear reactors and that kind of thing. Um, the the challenge though is not the bacterial capabilities. They're there, and we know how to use these capabilities in some way. Um, but then there are logistic limitations in doing it on a large scale, and so that I don't know. That I don't know how far we'll be successful.
Yeah, that's